So today we're going to look at antibodies, right? So in this learning outcome, you need to relate the molecular structure of antibodies to their function. So relating structure to their function. So I would like you to check again, when you learn about the immune response, we have talked about the humoral response, where the final product that produced is the plasma cells will secrete antibodies. And these antibodies are specific towards the antigen that actually stimulate the B cells to divide and differentiate. So these antibodies will bind to the specific antigen and mark it to be removed by the phagocytes, which are neutrophils or macrophages. So today we're going to look at the structure and function of antibodies. So what is an antibody? Antibodies are globular glycoproteins. So remember, what is the meaning of the word globular? Angela? Globular stands for what? Spherical. Spherical, yes, very good. Spherical, right? Globular is spherical. Good. It's a three-dimensional shape, right? Formed by extensive bending and folding of a polypeptide chain. Now, what about glycoprotein? What is glycoprotein, Adrian? Glycoprotein is what?
Is a protein.
Why is it glyco there? Glyco stands for what?
The lipid.
Lipid? No, cannot be. You know it's glycogen, right? So what is the monomer of glycogen? Okay, so someone typed the answer just now. Let me see.
The carbohydrates.
Yeah, carbohydrate. Yes, protein with carbohydrate. Correct. Glyco means carbohydrate, right? And then protein means protein, right? Protein. So that means to say that this antibody, the composition of this antibody is made up of two types of biological molecule, the carbohydrate and protein. Okay, it's made up of carbohydrate and protein. So antibodies are globular glycoproteins and form the group of plasma proteins. So plasma proteins are proteins that are found in which part of our body? Protein that are found in which part of the body? Open to the floor. Blood, very good. You know blood. Okay, protein found in the blood plasma is plasma proteins. All right, so found inside the blood. So it will circulate in our body, right? Antibodies, once secreted, it will circulate in our body. So it can circulate in our body means can circulate in the blood, tissue fluid, and the limb. All right. And it is also called immunoglobulin. That means globular protein that is involved in immune response. So this is the definition of antibody. Okay, right, next. So let us look at the structure of antibody. Right, so antibody is a glycoprotein, right? So let's look at the protein part. The protein part, it is made of 4 polypeptide chains, as you can see here. 1, 2, 3, 4. 4 polypeptide chain. So you got 2 light chains and 2 heavy chains, because there are 2 chains that are actually shorter, okay? And we've got 2 chains that are longer. So therefore, 2 light chains for the shorter chain, 2 heavy chain for the longer chain. Okay, so 2 light chains and 2 heavy chains. And all the chains are joined by disulfide bridges. So you can see the disulfide bridge here, right? Joining the two polypeptide chains, and you got two more disulfide bridges here that join these two heavy chains. Okay, one disulfide bridge joining the light chain and heavy chain. One disulfide bridge joining the heavy chain and light chain. Two disulfide bridges that join between the two heavy chains. Okay, very easy to remember. Okay, next, each chain has got variable region, a variable region. So you look at this part here. This part here is shaded darker in color. So this part here are the variable region of the polypeptide chain. Variable region means this region here can be different for different antibodies. We have got many different type of antibodies. Each antibody can bind to only one type of antigen. So why do we have got different type of antibody? The part that is different is actually this variable region of the polypeptide chain. So can you tell me specifically, how is it that this part of the chain can be different? Different in what? Different in what form? Different in what form? Can you tell me how is it can be different? Base sequence. So Lowenthal, is this a DNA that is got different base sequence? This is not DNA, okay? This is the what? This is a polypeptide chain. So what can be different that make it different? Different shape. Why is it different shape? Correct, it's different shape. So why is it different shape? Correct, Rafina is correct. Because it is made of different amino acid sequence. The entire polypeptide chain is made up of the monomer amino acids, right? Joined up by peptide bond. So when you say that this region is different, that means the amino acid sequence is different. So when amino acid sequence is different, that means you're saying that primary structure is different. So therefore, secondary structure, tertiary structure can all be different because of different primary structure. So the amino acid sequence is different. Therefore, this is also different. Therefore, this is also different. So that's why the shape can be different because of this. Amino acid sequence is different. Okay, so variable region is different because the amino acid sequence is different. Therefore, it gives an overall different shape here. So because of different shape, different antigen can bind to it. So each antibody is specific because of the specific variable region. Okay, so made up of different amino acid sequence, so therefore give rise to different tertiary structure, so it can bind to different antigen. So different antibody is different because it's got different variable region. Okay, so what about those regions that are not shaded? So the rest of the region that are not shaded, that means they are the same amino acid sequence in different antibodies. They are all the same. Okay, they are all the same. So that's why you remember this region here. So we can further divide the antibody into two sections. This region here is called the FC region, and this whole section here is called the FAB region. So two more regions. FC region is called fragment crystallizable. So this is the region where the macrophages will have got the receptor for this region here. So that's why regardless of antibody, as long as all the foreign microorganism has been marked by antibody, this region of antibody can bind to this receptor because this is a receptor for the FC region of antibody. So regardless of the antigen shape, this FC region of the antibody will all be the same. So therefore, your phagocyte only need one type of receptor here, which is receptor for FC, the FC receptor. Then it's able to bind to all sorts of antibody. Okay, it binds to the FC region of the antibody. They're all the same, regardless of different type of antibody. Now, this region here is called the FAB region. AB stands for antigen binding. So F stands for fragment. So the fragment means the part of the antibody which can bind to the antigen. So FAB region, because this region here, the variable region here, can bind to the antibody. Okay, so can you tell me one antibody can bind to how many antigen? One antibody can bind to how many antigen? Look at this diagram and tell me. 2, yes, correct. Annabelle is 2, right? Why 2? 1 here, 2 here. See, antigen binding sites, you got 2. So it's got 2 antigen binding sites, means one antibody can bind to 2 antigen. Now, I want you to look at the diagram again and tell me, where is the location of the carbohydrate? Because it's glycoprotein, right? So where is my carbohydrate? Can you annotate, please? You can annotate or not? I'm not sure you can annotate or not. Okay, cannot. Okay, can you see where's the carbohydrate or not? Can you describe to me the location of carbohydrate? I'm not going to call you all better. So can I have got Cheng Han, He Cheng Han, to describe the location of the carbohydrate chain?
It's on the long chain, the heavy chain.
It's on a long heavy chain, correct? The long heavy chain. Which part? FAB or FC region?
FC.
FC region here, right? Right, can you see here? Chain of sugar molecule. So this is a carbohydrate chain attached to the FC region of the antibody. So that's why the antibody is called glycoprotein. Okay, glycoprotein. Okay, so that's the structure of antibody. Now let's move on with a little bit more about the structure of antibody. There's one region which is called hinge region, and this hinge region is spring-like. Now, if you want to understand the meaning of the word hinge, right? So let's say this is your door frame. This is the frame of your door. Okay, and then this is your door here. Okay, I'm going to draw your door here. Okay, so your door can open and close because usually you have got these two pieces of metal that's attached to it that's flexible. So it allows you to open and close the door because of this region here. So this region here is actually called the hinge region. The hinge region of your door is flexible. It allows your door to open and close. So same as in the antibody also has got hinge region. So the hinge region is located here. Okay, so the hinge region is located here. That means to say that this is like the door that can open and close. So what is so important about this hinge region is it provides flexibility to the antibody to bind to antigens that are located at differently spaced. To bind to antigens that are differently spaced. That means these two antigens are very close. So this antibody can bind. If the two antigens are further away, it can also bind because it is able to open up further away to bind to that antigen because of the hinge region here that's flexible. Okay, so just like the door hinge. Okay, next. What about the FAB and FC region of the antibody? So FAB is a short form for fragment antigen binding region. Fragment antigen binding region. So it contains the two antigen binding sites. Different antibody will have got different antigen binding sites. That means the amino acid sequence here in the variable region will be different. Because of that, the overall shape here will be different. So because of that, different antigen will bind to this antigen binding sites of the antibody at the FAB region. Okay, now the second part or the second fragment of the antibody is called the FC region, which stands for fragment crystallizable region. So this region, phagocyte will have got receptor that can recognize and bind to the FC region of this antibody. Okay, so therefore different antibodies, they will have got the same FC region because FC region is the constant region. It's got the same amino acid sequence. So therefore, for different antibody, the FC region will be the same. So the phagocyte only need to have got one type of FC receptor. It can bind to all the FC of all different kinds of antibody. So the advantage is by having one type of receptor, the phagocyte can bind to all the different types of antibodies. Isn't that wonderful? It's like you've got a master key, yeah? A master key can open the door to all, it can open all the doors in your house and all the doors in the hotel room. Okay, so that's master key. Now, this is not in the syllabus, but it's actually for you to realize something. Now, remember we said that antigen is any big molecule. The keyword is big, right? Any big molecule that can be recognized by our immune system as non-self or foreign, and therefore that will activate immune response. So if it's big molecule, if let's say, for example, this is an antigen, it's a protein, it's big. So it can be as big as the antibody, right? So if the antigen is as big as the antibody, of course, the antibody cannot bind to the entire antigen, isn't it? Because it's the same size. So therefore, the antibody actually can only bind to a small section of the antigen, right? Like the diagram that's being shown here. It only binds to the small section of the antigen, like this part here, right? This part here, and this part here. Only bind to a small section. So this small section of the antigen where the antibody can bind to is actually called the epitope. So that's why you see epitope 1, epitope 2, and epitope 3, all of them got different shape because it's different section of the antigen. So therefore, when one antigen enters our body, it can actually initiate it can actually initiate different type of antibody to be produced against one antigen. So in this case, 3 different types of antibodies are being produced. That means to say you got 3 types of B cells that are being activated. Right? Activated means they undergo clonal selection, clonal expansion, isn't it? 3 different types, means 3 different clones of B cells can be activated here. So this is just to illustrate to you that antibody size could be similar to size of the antigen. So it cannot bind to the whole antigen, it can only bind to the epitope part of antigen. So that will cause more than one antibody being produced against one antigen. Okay, just for your information. Now, this part you need to know. How do antibodies work? So just now we look at structure, now we look at the function. Okay, so how does the antibody work? Many ways. It depends on the type of antibody. Okay, number 1 is to prevent entry. Why it prevent entry? Because when the antibody binds to the virus or it binds to the toxin secreted by the bacteria, it makes the entire structure of the molecules becomes bigger. So when it's bigger, it can no longer enter our host cell. So therefore, it cannot affect and act on our host cell. So it cannot infect the host cell. It cannot affect the metabolism in the host cell. So this is prevent entry. First mechanism. Second way is it prevent movement. So you can see in the diagram, the antibody binds to the flagella of the bacteria in this case here. So therefore, the flagella cannot move. So it cannot move properly. It cannot propagate the bacteria. It cannot propel the bacteria to move forward. So the bacteria that enters our body will remain in whichever location to be phagocytosed away. So it doesn't migrate to other parts of the body. Okay. Third way is agglutination. Now, agglutination, just remember, agglutination is glue. So it will glue all the bacteria together, as you can see here. So you can see this antibody is very special, right? it can stick to one bacteria on the left side and stick to another bacteria on the right side. So when you stick these two bacteria together, these two bacteria is restricted to move. So this bacteria to move this way, that bacteria to move that way, they cannot move already. They're all stuck together. So this is called agglutination. Okay, antibodies with multiple antigen binding site causes agglutination of bacteria, reducing the chance of them to move and hence spread to other parts of the body. So this type of antibody that causes agglutination is called agglutinin. Okay, now fourth way of the antibody acting is it will cause lysis. So that means when the antibody binds to the bacterial cell, say for example, then it will cause another molecule to come and punch hole onto the bacterial cell wall. So it can cause a lot of holes to be punched on the bacterial cell wall, and hence the cell wall is weakened. So as the water enters the cell, okay, the cell will continue to swell and burst. The cell wall is no longer rigid enough. Okay, so therefore, because a lot of holes are being punched, so therefore it can stretch until the cell bursts. So that will cause lysis to the cell. All right. So fifth way of the antibody working is by opsonization. Opsonization, this word stands for seasoning. So remember when you want to eat barbecue chicken wing, right? You season the chicken wing, isn't it? You season it with pepper, season it with what else? I don't know what kind of herbs you want to season it with. Okay, but you don't add honey. Don't add anything that is sweet, because if you add anything that is sweet, if you go at barbecue, it becomes char. You know, charcoal char, it totally burnt to become black color. You only add honey after you barbecue. Okay, after barbecue, you actually you add the honey onto it. Okay, don't add before, add after. So opsonization means seasoning. So what do you mean by that? Opsonization means the antibody will go and coat the bacteria like this. It's like seasoning the bacteria, making it more delicious to attract the phagocyte to come and phagocytose it. So this is called opsonization. Coating the bacteria to make it easier for phagocytes to ingest them, because phagocytes have receptor protein for the Fc region of antibody. So this type of antibody is called opsonin that causes opsonization. Okay, opsonin. Now the last way of antibody working is neutralization or precipitation. That means it will bind to the toxin of the bacteria or the viruses. It will bind to the viruses and neutralize it to make it unable to activate, make it unable to act on our body cells. So it's neutralizing it. Precipitating, P-R-E, precipitating it means that it will make it solidify, right? So when it solidifies, it will no longer act on our enzymes, act on our substrates and so on. So this is called precipitation, because when you talk about chemical reaction, a lot of reaction, you need the substrate to be in aqueous form, right? So now after binding to antibody, the substrates becomes solid form, so it cannot act anymore. Like for example, inhibit our enzyme, okay, it cannot act anymore, so it is harmless to our body. So this type of antibody is called antitoxin, because it prevents the toxin from acting on our body by neutralizing it or precipitating it. So therefore, there are 6 ways an antibody can work against the pathogen or the antigen. So let's recap the 6 ways. Number 1 is prevent entry. Number 2 is prevent movement. Number 3 is agglutination, clumping all the bacteria together. Number 4 is causing lysis. Number 5 is opsonization, seasoning or coating the bacteria to be removed by phagocyte. And number 6 is neutralization or precipitation. So 6 ways antibodies can act against the antigen, okay, or the bacteria or the virus. Now, how a bacterium enters a whole cell. Remember when you learn about phagocytosis, you learn that first is chemotaxis, right? The phagocyte will move towards the bacteria because of certain chemicals that are secreted around here. So what are the chemicals that are secreted around here? Can you give me three different kind of chemicals that can cause chemotaxis? Joey, 3 chemicals that can cause chemotaxis. Joey. No, Joey?
I'm not sure.
Jaswinder? This one is your quiz, right? You're not done your quiz for this week, is it? Your quiz homework. Every day after the lesson, you need to do it after the lesson. Cannot wait until last minute and do everything. Huh? You won't learn as well as if you do it every day after the lesson. Yes, very good. Crystal says histamine. Yes. This is the first chemical that's secreted upon infection, right? 2 more. Give me 2 more. One is from the bacteria. What is it? Bacterial toxin, bacterial component, Myra already said, very good. Bacterial component like toxin secreted by bacteria. One more, the third one you learned already during immune response was cytokine, right? Cytokine can be secreted by, if you look at the video, it says it can be secreted by macrophage, but it's not in the syllabus. You learn that the T helper cell, the active T helper cell can secrete cytokine, right? Okay, so these are the 3 chemicals that you have learned that can cause chemotaxis. So phagocytes will move towards the bacteria to phagocytose. So after chemotaxis, it must attach to the bacteria. You learned already. So you learn that there are 2 ways it can attach. Either it attach directly to the membrane, like this one here, or it can attach to the receptor. So let's say this is a marker. This is a marker, which is the antibody that attach to the antigen on the surface of the bacteria, okay, and it got a lot of antigen. So the antibody can bind to the FC receptor, and then after that, phagocytosis will happen. Or it bind directly to the membrane and phagocytosis can happen. So you have learned this way and you've learned this way. Now this one is a cell drinking process where the membrane have to invaginate inwards. Invaginate inwards, learn up this word, invaginate. 1 word, 1 mark. Okay, learn it up. Okay. So it's showing you how bacteria can enter the whole cell. Bacteria is solid, so therefore it's by this way phagocytosis directly, or receptor mediated endocytosis, means it has to attach to the receptor, then only enter the cell. Okay. So this is by phagocytosis as well. Now, how a virus enters a whole cell. So this one is recap HIV virus. How come HIV virus only enters T helper cell? Right? It's because T helper cell has got a receptor on its membrane, which can bind to the glycoprotein found on the surface, found on the envelope of the HIV virus. So therefore, it only able to bind to the receptor found in T helper cell and therefore only enter T helper cell, convert into viral production factory, right? Okay. So there are 4 classes of antibody, but it is not in your syllabus. So therefore you don't have to learn this, okay? You don't have to learn this. All these 4 classes you don't have to learn. So therefore I'll skip this, shall we? Okay, I'll skip this. If you're interested, you can read this or else you just skip the whole thing, right? Now, let's look at this graph. Let's do partial question 37. The graph shows the amount of antibody produced in response to an antigen, right? An antigen. So this is the concentration of antibodies in the blood. And this is time in the number of days. So this is the first exposure to the antigen. So day 0, antigen enters the body. So you can see that for 5 days, there is no antibody that can be detected in the blood. Only after 5 days that the antibody concentration start to increase. So from the graph, which statement is correct? Okay, I give you time. Please type your answer. Myra is very fast. Your check. What is the answer? Please type your answer. Okay. Only Myra type. The rest of you? Mahi Jaiswal says same as B. Okay, let's see the answer whether it's correct. It takes 25 days to achieve active immunity. No, because you see, by day 5, antibody already increased. It cannot be 25 days only achieve active immunity. Instead, at day 0 already got active immunity, because day 0, after the antigen enters the body, it will first start by phagocytosis. After phagocytosis, you go and present the antigen already, right? Then after that, it will select the T helper cell and also B cell. Then after that, clonal expansion of T helper cell, divide, differentiate into active T helper cell and memory cell. Active T helper cell secrete cytokine. So it activates the selected B cells to undergo clonal expansion. So the cell divide and differentiate into memory cells and also a plasma cell. So plasma cell will then secrete the antibody. So therefore, only day 5 onward, you can detect antibody, because day 0 to day 5 is when all these processes happen. All these processes here happens, right? Phagocytosis, antigen presentation, clonal selection, clonal expansion before antibodies being produced. That's why this is called the, what is this period called? Can you please type your answer? What is this period called? Hello? This period. Incubation period. No, it's not called incubation period. What is this period called? Delay. Window period, correct. Myra, correct. This is called the window period. If you have not learned it, please learn it now. Window period. Why my window period like that one? Okay, window period. Okay, so window period is a period of time where the antigen already entered the body. Hello, the antigen, okay, enters the body already, but no antibody is produced yet. So that's called window period. Why is there a turn there? What is turn? What got Chinese word there? Okay, window period. The time when the antigen enters your body until the antibody can be detected is called window period. Okay. Now, what about just now someone said incubation period? So what is incubation period? What is it different from? How to type? Okay. They say incubation, right? Incubation period. Incubation period is totally different from middle period. Can you tell me what is incubation period? Let me call someone to answer. Vana, what is incubation period? What is incubation period?
I'm not sure.
Okay. Vana, not sure. What about, I'll call someone else. Chok Yiman, what is incubation period? When you learn infectious disease, 1 whole week, right? You definitely come across this word incubation period, right? So when you come across and you don't understand what you need to do, find out the definition, right? Okay. So what is incubation period? Delay. isolation? No lah. Someone say what? Isn't it the period until you see the symptoms from when you're infected? Yes, Mahik, you're right. Incubation period means after the pathogen enters your body, right? Until symptoms and signs starts to develop in your body, that period of time is called incubation period. After the pathogen enters until you get sick, until you can see the signs and symptoms, that's called incubation period. So that's why incubation period is not the same as window period. Window period is until antibody can be detected, you know. Whereas incubation is until you get sick. That means signs and symptoms can be seen. So they're totally different. Okay, so it's good that we clarify your doubts today. You need to know each word, what does it mean, and you use it in the correct context. All right. Okay, so this incubation and this is window period. So both are delay. Delay in what? You have to say. Okay. Right. Good question. B, memory cells for this antigen are present in the body within 20 days. Let me erase this out so you cannot see. Correct. So within 20 days, the memory cells can be seen. Yes, it's correct, because on day 5, when the antibody is produced already, the moment antibody is produced, that means plasma cells produced. If plasma cells is produced, because antibody comes from plasma cells. Okay, and if plasma cell is there, that means the memory cell is also there, because they are produced together by differentiation, right? Memory cells. Differentiation from the selected B cells. Okay, so you have to move backwards. So what about C? T helper cells are activated on day 12. No, it must be before that, because T helper cells are activated even before selected B cells divide and differentiate, isn't it? So it's before that. So this is wrong. Last one, the second exposure to antigen occur on day 25. It cannot be day 25. Day 25, the antibody level already increased. Right? Day 25 is here. Antibody level already increased. So it must be somewhere here. Right? Somewhere here or here. So it's before day 25. Okay, next question. 37. An enzyme hydrolyzes the 2 heavy polypeptide chains of an antibody molecule. The hydrolysis occurs at the hinge region and breaks the antibody into 3 fragments. How many of these 3 fragments are able to bind to antigen? Okay, type your answer. Myra is very fast. Huh? C, correct. Now you need to be able to draw the antibody structure. Huh? Okay, so if I cut it, I cut it here where my disulfide bridges is here, right? Disulfide bridges is here. So I have got 3 fragments. I've got 1, 2, and 3. So out of the 3, these 2 can bind to the antigen. So answer is 2. Okay, right. Question 36. What is the function of the plasma cells during an immune response? Very easy, right? This one, to secrete antibody. So this is a free gift in MCQ. Usually MCQ got free gift one, like this kind of very direct question. How many very easy question? Usually 5, I think. 5 very easy question, 5 very difficult question. The rest are average, I think. So usually when I set MCQ, I'll set 5 or 6 extremely difficult question and 5 very extremely easy question. If the intake, the students are really, really good, I might set up to 10 very difficult questions. Usually it's very rare lah. Usually we set average. Okay, let's do this question from the textbook. Explain why polysaccharides would not be suitable for making antibody molecules. Now, this kind of question can come up in the actual exam for higher order thinking question. Instead of explain, they will write the word suggest. Every time when they ask the word suggest, it's usually a higher order thinking question really. So can anyone answer? So let's look at the answer. Polysaccharides are made up of only a small number of different sugars. That means different monomers. Unlike proteins are made up of 20 different types of amino acids. So therefore, it will be able to give a huge number of different molecular shapes in the variable region of antibodies versus if we use polysaccharides. So you see the answers are very specific. The keyword that they will give mark is actually you need to state there are 20 different types of amino acids that are possible, but a smaller number in the polysaccharide chain. Therefore, it will give a huge number of different shapes of variable regions. So they want you to write about variable region because this is the region that is able to bind to different antigen. So that, you know, in the entire lifetime that you're alive, right? You can be living for 75 years, 85 years, 95 years, probably 100 over years old. And then within your lifetime, you'll be exposed to so many different types of antigens, right? And whatever different type of antigens that you're exposed to, your body must be able to produce antibody against it. So which means to say that it must be able to produce all sorts of antibodies, right? It is above like millions, right? Millions of different kind of antibodies it should be able to produce. So therefore, it must have got different kind of amino acids, many different amino acids, so that it can give you all sorts of different primary structure, so secondary, tertiary, quaternary structure, right? So this is a very good answer here. All the keywords are here. Now, the next question is, explain why only some B cells respond during an immune response to a pathogen. So only some B cells will respond during immune response to the antigen. Okay, who would like to answer? Why only some? Why not all? This one very easy. Okay, I'm going to call someone to answer. Okay, can I have got Geng Yiwei? Geng Yiwei to answer.
I'm not sure.
Huh? We just learned the immune response only. Remember? Clonal selection. Do we select all the B cells? Which B cells will be selected? Lo Wen Tao asks, mast cells are found in all the tissues in the body, okay? Yeah, Martin, correct. Only some B cells have the specific antibody receptors. Yes, correct. So this one is for you to remember here. So that's why this one you have to learn up. Huh? We learned that during immune response, you see, you got 3 different clones of B cells that are being shown here. Out of the 3, only B2 cells are selected. So they undergo clonal selection because only B2 cells have got antibody receptors that can bind to the antigen that's presented by antigen presenting cell. So that's why among all these 3 clones of B cells that are different, they are different because their antibody receptor got different shape. Only B2 can bind. So therefore, only B2 is being selected. Okay, so with that information, you will be able to answer this question here. Where is the question? Stop share. So I have to share again. Huh? Okay, right. How to share again? Share. Okay. Okay, come. Nice. Okay, so let's look at the mark scheme. Only some B cells and T cells have the receptors of the correct specificity. That means only some B cells and T cells, B cells having antibody receptor, T cells having T cell receptor, which is able to bind to the antigen. That's why they call specificity, means specific in terms of complementary shape. Okay, right, so very good question. Why am I scrolling here? Okay, last question. There are many different strains of rhinovirus. So you know that rhinovirus will cause flu, right? Rhino means rhinoceros, the rhinoceros. Yeah. Okay, rhinoceros. Okay, there are many different strains of rhinovirus which cause the common cold. Explain why people can catch several different colds in the space of a few months. So that means to say that when you catch the cold, right, your immune system is supposed to have been activated so that the next time when you get the same virus, your body will remember that virus. So therefore it can launch an attack towards that virus very quickly to remove them. So therefore it will not cause illness to your body. That means it will not cause cold. So this question is saying that how come after you get a cold, you will still get a cold in a space of a few months? So who would like to try? Like, you know that you get cold when you're a baby. After that, when you're in kindergarten, you still get cold. Primary school still get cold. Secondary school still get cold. Now in college also can get cold. Why can still get cold? Martin says that because we only have immunity against one strain of the virus. Different strains of the virus have different antigen. Very good. So there are so many different strains of virus that can cause flu, isn't it? So say, for example, now this particular strain of virus enters your body, right? So the antigen is, let's say, this shape. Now, next time another virus enters your body, the antigen is triangular, right? Next time another flu virus is a tiny little flu virus enter, the antigen is like a straight line, right? So every time the antigen that comes in is different because it's different strain of virus, different strain of rhinovirus. So rhinovirus is actually a general term to the virus that can cause flu. And this rhinovirus got many different strains, right? So therefore, a different strain has got different antigen. So it will activate a different antibody to be produced. So therefore, your body is not exposed before to the antigen. So the immune response will be slower because never seen before. The antibody concentration produced will be low. Okay, so first time when your body is exposed to the antigen, right, your immune response will be slower and also the concentration of antibody produced will be lower as well. Okay, so can you see that the question in your textbook, they are really, really good because it brings you to actually think further, think deeper. So this is when you try out this question, right? It will prepare you to answer those higher order thinking questions, especially those of you who want to score A star. Okay, so if you want to score A star, this will be the place where you want to look at the questions to train yourself to think deeper so that you are able to answer all the higher order thinking questions in exam. All right, so therefore, let's recap what we have learned in today's lesson. We have looked at the definition of antibody, which is a globular glycoprotein, and it is an immunoglobulin. And you look at the molecular structure of antibody, which is made up of 4 polypeptide chains, where 2 are the light chain, 2 are heavy chains, and all these chains are joined by disulfide bridges. Light and heavy is by 1 disulfide bridge, whereas heavy and heavy is by 2 disulfide bridges. Then there are 2 antigen binding sites for the usual antibody. So that sites, the chains are variable region. And then we also can divide antibody into FAB region, antigen binding region, and FC region, crystallizable region. Then there are 4 classes of antibody, which are called immunoglobulin G. So IG stands for immunoglobulin. Okay, So we've got IgG, IgA, IgM, IgE. This is not in your syllabus. What I want you to learn is Ig stands for immunoglobulin, means antibody, right? So 6 functions of antibody. So different antibody will have got different function. So it can be preventing the entry, prevent movement by attaching to flagella. Agglutination means will attach to multiple bacteria, causing them not to be able to move, and they will glue together, causing lysis. Opsonization means seasoning, coating it to be removed by phagocyte. Neutralization means render it becomes harmless, right? Neutralization. So that's the lesson for today.